Yes, so my aim for this afternoon is to do a deep dive of our connectivity atlas, the cell type database, and the brain observatory data. And my intent is to give you some sense of what that data is, what we did with it, and how we package it as a product, and how you can access it, especially for your projects or when you go home and do further research. I have more slides than we have time for. So also ask me questions, and then we are also going to walk into the web app as well, so that you can see visually what I'm talking about. I'm going to skip this one. Okay, so as Christoph says, all our data is available through our Allen Brain Atlas data portal, so it's brain-map.org. And then in particular, we're going to concentrate on these three atlas. You click on the big buttons, and it will take you to that part of the product. So our Allen Mouse Brain Connectivity Atlas essentially is each data set is axonal projection, images of axonal projection from a selected neuronal population. And why we want to do that is that we wanted to end up with a quantitative way of computing a mesoscale connectome. The second product we're going to talk about is the Allen cell types database. Currently, it doesn't actually have the cell typeness of it. Currently, as it stands, it's basically a multimodality characterization of single cells. And again, quantification is an essential part of this, because eventually all of that data is going to get wrapped up so that we can do a data-driven cell types taxonomy. So this is what we typically do. We'll release data when we have generated it, so that we're doing analysis, everybody else is doing analysis, but then that gets summarized up into some sort of additional framework or knowledge that we also then wrap up into our product. And in July this year, we released our Allen Brain Observatory. So in this product, we're basically now doing surveys of activities of cells in awake behaving mice. In this first release, what we're trying to do is the mouse is on a wheel running around, and it's looking at visual stimulus, passively looking at visual stimulus, and then what we're doing is then correlate the activity we see with the stimulus, so that even on the website you get some sense of the what are the function of the cells, what kind of tuning properties the cells have, is it tuned for running, that sort of thing. So, okay, and then also with the data with our product, there's actually three different components to our product. There is the web application, and our intent there is that you can go online and you can search and browse through our big data set to find what you need. We have an API, so that because it's a large data set, you most likely want to script and download the data. And as you know, for the cell type database and the observatory, the metadata is very complex. So within the last couple of years, we've launched our Allen SDK, which is a set of Python tutorials and scripts that's basically trying to help you get started with your analysis. So it calls our API so that you can get data into a data frame that's data frame that you can use more quickly to do your analysis. So
terabytes, petabytes, gigabytes.
There are orders of petabytes. I think there are petabytes of data. I think all of that together is 3 petabytes data. But again, you might not want data at the raw level. So that's what we try to do here is there's data at the raw level, but typically what people want is the quantified data. And by the time we get that, you have this huge data reduction, and those data sets you can easily download. But the people who want to do the raw image processing, then they need to figure some way of how to deal with that large data.
Disks, so we've done that. Because each experiment, gives you hundreds of terabytes. So at some point you can't go with net anymore. Right? So if people really want all data, we ship them with this. You ship us this with data.
You ship us a disk and we will give you data, yes. Okay, let's start with the connectivity atlas. So when you press the button, you'll land on this page. And what you see here is a big giant map, and each dot represents an injection site. In our current release, there's about 2,300 injections. So at each injection site, a virus is injected into the brain. The cells that get infected, their cell soma, their axons, the dendrites, the local dendrite, all fluoresce a green color. And that is the green signal that we're trying to image and then quantify to get a quantitative readout of projection. Okay, while we call this the connectivity atlas, and we sort of map everything into one common framework and one common database, there are actually different flavors to this experiment that you sort of need to understand when you dive into this data. Okay, so the first subset of this data is injection into wild type, but that's actually C56 black 6, so wild type. And then we do about 500 injections spanning the entire brain. Each of these injections are kind of relatively big, and it's intended to be biggish. And in the cortex, actually 2 injections are done so that you span the whole cortex. So the intent of this subset of data set is actually to provide a core scale brain-wide connectome. So that you're covering injection input at every part of the brain, and because we do 3D imaging, you get an output for the whole brain. And then this was actually the data set that was used in our Nature paper. So there was only talking about the wild type data set in this paper. And there's some of the visualization that we did with this data set. All right, so then the next phase of this project is to start to use Cree lines. So in the subcortex, you would want to use Cree lines to target small structure that you can't target just by doing just a coordinate injection. So the scientific team look for Cree lines or genes that are specific markers for certain small nucleus, and then those are used for the injections. That's why there are a lot of data in this part of the application so that you can target small structures, and also to target subpopulation like interneurons or parvalbumin or SST as well. In the cortex, the Cree line is actually then used to target different layers of the cortex. So then, so for each different Cree line, what the intention here is then to try to target the same cortical, not just the region, but the cortical location, so that when you're injecting mice for the different layer, that there's some way of that you can recombine the information so that you can see how different cortical layers project to different parts of the brain. So when you look at these injections, they tend to be grouped into these little pockets, so that I think they were covering 30 different areas, so that you have 30 little columns so that you can actually then start to pass things out in an interareal cortical connectome. And another component of the data, 77th injection was actually done in the retina because we're interested in vision, so we wanted to understand the central projectome of the retina. So on these data sets, the injection was done such that, so in particular Cree lines that enriched for particular, I think it was just the ganglion cells. And then and what they tried to do was to make sure that the injection covered the whole retina. So this wasn't a project to try to understand topography. So the injection made sure it spanned the whole retina. Our imaging is actually just in the brain. So then we used the optic chiasm as the starting point of this data set. So So for people who are interested in the retina projectome, then this is what this data set is, and whereas currently our scientists are trying to then collect this data to see where the different cell types are, where they project to in the brain. And then two of our newest data set is then looking at inputs and outputs from the visual area. What we have done in this data set, it's also to incorporate intrinsic imaging. So before the injections are done, the mice is anesthetized, and now looking at drifting grading stimulus so that we can find the different, compute, find the phase maps compute the phase maps of so that we can compute the different visual areas. And this is done for two purposes. The first one is that we want to inject into a functional area. So now if we want to inject in AM or PM, then we're actually using the functional information for targeting. On the reverse side, when we're injecting in subcortical areas, we can then interpret the projection into cortex in terms of the functional area as well. So then this is why it's important to integrate the functional information with the projection information. And then for this part of the project, actually two different viruses are used. The first one is the GFP that basically it's expressed all through the cell. And then we do a side-by-side with synaptophysin, which is just targeting the synapse, and then to compare the differences in the quantification that you'll get with and without basically the axon as well. And then in our fall release of this year, our additional data set is then so when you inject in one cortical area, you are going to get a population of cells. These cells, each cell, if you see projection to two different areas from this experiment, you can't tell if every cell goes to both areas or if each cell just target one particular area. So our new data, the data set that's coming in the fall release is actually it's target defined. So there's actually going to be two injection sites. The first injection site is actually a retrograde injection. So when you are injecting the virus here, only cells that are projecting to the first red injection would be infected. So this then allows you to break down the cell population even further down by where it projects to. And then targeting for 2018 release, that all the data so far it's anterior grade projection. In the 2018 release, then we're looking at retrograde proper connection because it's a rabies-based virus. Okay. So then this is, so in one data set, when you do injection, the data looks like this. So then we use a tissue site imaging system in which we scan the image on the block face using two-photon microscopy so that we're actually imaging a little bit down from the surface, and then we cut, and then we image again, and when it cut. What this gives us is that because you're imaging on the block face, you don't have deformation. So when we come to reconstruct this volume, then you have a really beautiful, pristine 3D volume. Because with connectivity, we care about things that are so tiny that this reconstruction and quantification, the accuracy of that is very important. This is compared to, for example, when we do our gene expression in situ hybridization, it's really difficult to recover from each deformation when you're putting things on a slide. So then this data set allows us to firstly reconstruct things very precisely in 3D and also allow us to register everything together. Because to actually make our connectome, you actually have to use the entire data set in concert. So then every data set goes through our processing pipeline. Well, in this data set, we actually start from tiles. So each tile needs to be stitched and aligned, flat filled, stitched back together. And then we then do a image segmentation for every pixel, classify whether this pixel is background or it's a signal. And for this data set, in both channels, there's a fair amount autofluorescence. So a lot of tricks needs to be done to figure out, are you background or are you foreground? And typically, while we're doing segmentation in the green channel, we have to actually use both channels to determine what the background is, because the background sort of varies throughout the brain as well. And then we register every data set to our common coordinate framework, which I'll talk about in just a little while. What that will give us is that so our annotation, our common coordinate framework comes with detailed annotation of the 3D brain at a 300 structure level plus layers of the cortex. So if we're able to register our data in this common framework, then we can actually parcellate any of the signal that we see from the segmentation into a quantitative readout. So once you get the data through this pipeline, we're able to visualize everything in a common 3D framework, but you can also get a structural readout of the projection signal in the different areas, and also quantify left and right hemisphere. So this is the data then is used to construct that connectivity matrix that Christoph showed earlier. So the beauty of this, the tissue site system, as I said, gave us these beautiful 3D data sets that we can really do a good job of aligning the data. So at the beginning of the project, all we had was the Allen Reference Atlas, because that's where we started when we did gene expression. And the Allen Reference Atlas was a single Nissl specimen in which we cut everything it was cut at 25 microns. But then the annotation in the reference atlas that you see was done only every 100 microns. So while we can reconstruct the Nissl in a sort of nice, smooth fashion, we had issues of these drawings being done in 2D and being very far apart. So even if you can get registration to work really well, the quantification still, you can still be out if you didn't have a smooth 3D structure. So, but this was then, but this is what we used at the beginning when we had our gene expression atlas. And then once we started to do our connectivity, the original atlas was also only annotated in one hemisphere. So when we started this project, that we knew, okay, for connectivity data, it's important to see the projection on the other hemisphere as well. So the first step we did was then, in version 2, we took the brain and artificially made it symmetric, so that now we can get quantitative readout on both sides. So we're closer, at that point in 2011, we're closer to having a common 3D framework that was used for quantification. But then when you came to wanting to register the autofluorescent signal from the connectivity data to the Nissl, we find that because when you want trying to do image registration, the closer the two modality of the two things that you want to register, the easier time you have, because it's a cost function. So if things are the same data modality, then it's easier to optimize just a transform. But if they're two different modality, you're optimizing that mapping function between a modality as well as the deformation field. So as part of that project, we made an average brain so that we will register everything to the average brain before we register it to the Nissl. But over time, we found that the average brain itself was actually giving us a beautiful 3D volume. So then in 2015, we decided to switch from our single Nissl brain to our average brain, and then started a 3-year project to annotate this 3D brain, so now we can have a true 3D common coordinate framework. So then to actually make our average template, it's actually a multiple step template. So because at the beginning you didn't have a 3D space, a smooth 3D space to start off with, you got to iterate that process. So you start off with our 1,675 specimen. You do a fine registration just to your Nissl, because our Nissl is the space that we've been with for 10 years or so, and we actually wanted to keep that sort of that orientation of the brain. But once you have registered to everything together to this first Nissl and average that becomes your first, our first average template. That is now, because as an average is now in the, it's got the same intensity as the autofluorescent, and then you iterate that again. And each time you do that, your average gets more clearer and clearer. And then you, once you've done that for a little while with an affine transform, then it's time to allow a little bit of deformable transform, and you let that converge, and then the next step you allow more parameters in your deformable transform, and you let that converge. And what you get at the end is then average signature that's sometimes easier to see in average than it is on a single experiment. So here we're pointing at the barrels of the mouse cortex, which comes out very clear in the average template, but it's harder to see at each individual experiment. And then in the hindbrain here, actually our person, our registration specialist, first pointed this out. What's the little spirally thing at the back of the brain here? None of us actually knew what that was, so we went and looked up what this is. So these are the barrelettes in the hindbrain, and each of these little subcomponent here actually maps onto each individual barrel in the barrel cortex. But in an individual experiment, you can't see that. But when you are able to do this registration over a large population, you can actually start to pull out signature that you can't see in a single experiment. And because of this, then we can also start to annotate structure that you can't do in a single experiment as well. So then in our May release, our analysts went in and then annotated everything they could see directly from the average template by itself, because that's easy. Just draw what you can see. And then they also started to do that 21 cortical regions here as well. So in order to get this, the visual cortex, actually I'll leave that in just a sec. And then for this fall release, our anatomist has finished out the cortex, and then we also have cortical layers, and we're releasing that data so that we can enhance the quantification of our cortical projectome. And at the end of 2017, we hope to then complete our annotation so that we can release that back. Because each time we have a new CCF, we have to go back and basically reprocess everything. So it's not something that we want to do very often, but it's essential to actually get this quantification, especially in the cortex, as accurately as possible. Earlier in the day, Christoph mentioned that, the cortex really is just a flat sheet of paper. And the fact that we want to use a mouse, because a mouse got a fairly smooth cortex. So when we started our next 10-year plan, and the plan was looking like we were going to image cells at certain layers, we're going to try to integrate information across different layers of a cortical column, it was also important to us to build an infrastructure that will allow us to integrate information across the layer, across each different layers. But because the cortex is actually bent, when you look at the data in a coronal sense, you actually, while people would draw a line to say this is the cortical depth and this is a corresponding column, it is actually not true. So what we would try and do here is then use a Laplacian to try to follow the shape of the brain, so that when we have information at one layer and then we want to integrate information on a different layer, we're going to follow, we compute the Laplacian, and then we actually walk from every point at the top of the brain, follow the Laplacian down to the white matter. And so that we have these 20 million streamlines that we can use then to integrate data. So this was important here because of how we wanted to annotate the cortex. So to annotate the cortex, we actually used projection data to annotate the cortex. So by the time we got to this project, we have 20-30 injection into the primary visual area. And with this data, we could actually start to see the topography of that data into the other higher visual areas. But then you have that, first you've got to integrate the multiple data sets, but then you need to sort of push that up to the cortical surface in order to do that drawing so that you can push it back down again. So the cortical coordinate system was important for us to do an annotation, but it's also important for us to actually start to visualize what's happening in cortex and to combine multiple data sets together. All right. So let's go back to the connectivity pipeline. So every data set that comes through, we do our segmentation, and then the next thing we do is actually then divide the data set up into little voxels. And then each of these voxels, we would compute a local statistic in that voxel to compute the average projection density, the total sum of fluorescence signal we see there, and then also we quantify injection sites also within each voxel. And the reason why we want to do that is then our data it's easier to then resample this data into a common coordinate framework versus trying to resample images in that framework, or to move the CCF onto each image, each of the images. So by doing this, basically, then we got the data, we quantified it, and it's become these voxels. Every voxel is in a common space. So now to compare any location between multiple data set, it's just the same voxel, so you can easily do things like correlation search and target search and injection search. So this is how we get all the data in the same space. These voxels on our web app, which I'll show later, we actually read all the voxels into our server so that you can pick an experiment, and then you, by comparing voxels, you can do correlation between multiple data sets. So these voxels are part of what we also provide through the API. So as I said, we had used these voxels on the web app to provide some searches, but in our Nature paper, we used the voxels to actually try to quantify how much topography or the relationship, how much of this topography between the cortex and the striatum and the thalamus, and the relationship between different areas, and that was also all done in a voxel-based way. In that original paper, you saw the linear model or the network analysis was done in a structural rolled-up view. But more recently, then with our collaborator at University of Washington, these network analysis are now being done in a spatial voxel sense, so that you can capture more information like topography as well. And then this data then is also available for you to do projects on to look at local connectivity. Okay, so once you've done the segmentation and the registration, then you can quantify data into your different structures. And then on the web app, we lay it out like this. I will actually give a demo of that in a little while. And then you can actually pull the whole thing together to get a brain-wide connectivity matrix. All right, and then for the cortex, for us, it's, and when we're interested in cortical connection, looking at the brain from the top view helps visualize a lot of that information in an easier sense than trying to get that information from a coronal view. So this is also available on our website. And then for each of the data set that has intrinsic imaging, we've also registered our intrinsic imaging data also to the cortex, to the surface of the cortex, so that this is how you're able to, as a project, integrate functional information with the projection information. Again, we make all this data available first, and then we do our own analysis later on. All right. So then our database effectively, when you want to use this data, you can access a high resolution images. We have an image service for you to get whatever bit of image you want at the different levels and a different region of interest. You have access to all the gridded data so that you can form your own quantitative data structure at a spatial level. You have access to the CCF. The entire CCF and the annotation as we have it today is also available for use. And then all the data that we've summarized to structure can also be downloaded as well. Okay, so I am going to launch the atlas. So basically, when you come into the connectivity atlas, you can click on each of the injection sites. Here you go. What you see here is actually a 3D visualization of our gridded data. You can spin that around. And one of the things that we wanted to do is that it was important to be able to take people back to the primary data. So from our 3D thumbnail, you can always click on the thumbnail to update you to see what are you looking at in 3D, because sometimes the 3D is quite abstract, so that you can walk into the primary data. And then here is where you can get that structural summary for every data set. And it also allows you for every signal here, for every structure here, it can update you to go back to your structure of interest, and so that you can see that quantification. And then there's one more thing I would like to highlight. So far we've seen, when you're in the context of one experiment, how you can look at one experiment. As I said, we actually read the entire thing into our database so that you can actually query the data in a reverse sense. So that's right. For example, here, let's make this go. Let's turn this sideways. So here is our spatial search, so you can pick any point, say, in the thalamus. And then what this is doing then, it will go to the database and go find every injection that projects to this point. And then, so that's what comes up down the bottom. And then when we first did this, it's hard for people to know what this looks like. So then for every point, we also walk from the injection site back to your point of interest through all the high signal voxel. So the intention was actually using all the data set to give you a virtual retrograde projection map. If you had enough data, this is what it might look like.
You're actually doing a real retrograde projection map as well now.
Yes.
Simulated.
Yes, this is a simulated one because we didn't have that data, but then this is allow you to walk the data back, so to say, well, who projects to me?
Question also, so when you're injecting, what viral vectors are you actually using? Is it AAV?
It's AAV.
Do you know the serotype?
Do I know the serotype?
Can you just-
I believe it's in the white paper.
Okay,
Yeah.
thanks.
So that brings a good point that whenever you're in our app, if you click on documentation, that is a very full, lengthy description of everything that was done to create the product. And then if you get lost in the product itself, you click on help. There's also help of what each button do and how you search for things and how do you filter out different experiments. All right. I am going to then switch over and talk about cell types. Is there other questions on connectivity? Oh, there's one more thing. Okay. Let's go to the SDK. They're going to let me do this. So the link to the SDK is in this PowerPoint. So then here's basically a Python, an IPython notebook that to show how you can use SDK to download a lot of this connectivity data. In this first part, it sets up how you can talk to the API, and then how you can then download the experiments that you want. And there is there it is. There's the IPython notebook. It walks through examples of how you can pull out the structure summary for each experiment. How you can then create your own little connectivity matrix. And I think more importantly, it sort of teaches you how to use the grid data and how to compare it to the CCF. So then this bit of code has downloaded the gridded data, show you how to slice and dice it, and how to compare it to the Common Coordinate Framework. Okay. So now I'm going to switch over to the cell types database. When you come into the app, you, as Christoph showed, you see a lot of dots in a Common Coordinate Framework. Each dot represents a cell that.
Just a question related to volumetric data. Do you have a universal format like GIFT, so that you could, for instance, superimpose MRI data?
So all our data is in an NRRD format, and it's a format that's kind of popular in the imaging community as well. It was just something that didn't need a library. We had there was known libraries that can open in Python and MATLAB and C++. So we chose that so that it was a lot of people can access, but a lot of viewers that can read any MRI can read that as well. Actually, why don't we switch over to the app? I think it's easier. Let's just do types. All right, so this is our cell types database. There's two components of it. Currently, we have cells in which we have characterized its physiology and morphology, and separately, we have cells in which we have characterized its transcriptomic. Moving forward is something that we want to integrate together. So when you click on the physiology and morphology, you get basically a map of all the cells that we have currently surveyed. If you click on one of these cells, it would then highlight it in the table. Here is then basically a summary view of the electrophysiology properties, and this is a one action potential that will let you to look at the shape of the action potential, because that's one of the main electrophysiology characteristics, and then also a thumbnail of the morphology if we have one for that data set. Multiple Cree lines were used to survey this data, and in this little table, each Cree line is marked as whether it was predominantly excitatory or predominantly inhibitory, and also which layer it is enriched for. So for example, here we can click on SCNN1A, which is a layer 4 marker. And now we have reduced the data set to just cells from this Cree line. The other thing we were doing that we were surveying both Cree positive and Cree negative cell as well. So then if you only want it to be Cree positive, you turn off the Cree negative. If you also want to reduce the data to cell that has morphology, you can click on that. And then so now you can actually look at the different, all the cells from that Cree line to see how common it is. And then you can click on this. And then this brings you basically to our primary data for the morphology. You can actually look at it in 3D. You can zoom around, rotate, go in and out. The reconstruction was done from an image stack. What we're showing here are two minimum intensity images of the morphology, just to give you a summary view of that data. You can actually go and browse through the image stack as well to see the primary data, the primary biocytin data. If you go back to here, if you click this thumbnail here, it will take you to the electrophysiology. Here you can actually see every stimulus that was applied to this data set. Here we're looking at the long squares. So if you click on this, you can look at a single trial at a time. So in the long squares, we started with a current at minus 110 picoamp, and they then sweep through and increase that current in steps, and then you see that the response, the voltage response of the cell also increases with the current until it reaches a certain threshold, and then the cell starts to spike. And as the current increases, the spiking also increases as well. So as well as showing you the traces, we have also computed things like the resistance by looking at relating the injection current to the output voltage, and then looking at the spiking frequency as well. So, and all of this was summarized as features of the cell. And then when we look at, for example, the short square stimulus, the intent of this stimulus was to just invoke a single action potential. So then you can use this little widget here to just focus onto this data. And then you can also then also see the single action potential. So then for every one of these spikes, we also compute the upstroke and the downstroke, because that's another key signature of the cell. We also compute values like how much further down the downward trough here. This cell doesn't go down a lot. And for every cell, all of this information is then put into our database so that you can actually search the cells for different electrophysiology properties. And then all of this data, it's also available through the SDK and API as an NWB file as well, so that you can do your own analysis. So I'm going to take off all my filters. So this little parallel coordinate plot here, for each of these dimensions are actually the top 5 most sort of distinguished characteristics of the electrophysiology that our analysis team has computed. All the cells are actually color-coded by this upstroke downstroke ratio. Cells up here, we can then filter for the cells up here. Are the slow spiking cells that tend to be your excitatory cells, because if I only put the morphology on there, you see these are all the ones with your apical dendrites. And then if you move down to the other part of the spectrum, you get to your fast spiking cells, you get to your which are your interneurons. And then to look at the models, you can also filter for cells that have models. Here I'm looking for cells that have glyph biophysical models, both the local and all active models. And then you can go and find one and then open it. Open this one. And then you will find the models here. And what we wanted to do with the models was that we took the model and then we actually replayed the experiment back to the model and also show the output of the model at the bottom of the screen here. So here is a glyph model. The intent of glyph was just to match the firing rate, so it wasn't trying to match the action potential at all. But so here you're just try to comparing the difference between the spike raster down here, and then you can go back up here, and then look at the different levels of lift models that you see here. As the lift model becomes more complicated, it has increased explained variance ratio, or then you can actually click on the biophysical models, which should have a better match of the shape of the response as well. Okay. And then just like the connectivity atlas, if you go cell types and you go to the SDK, there's also another notebook that will help you get started to access the data, to plot out the traces, to actually filter the cells with the different metadata or the different features that we've computed, and to also access the morphology so that you can plot them and do your own analysis. Any questions on the cell types?
So if you have similar data to
Sorry, can you repeat your question?
Do you have similar data to the one that you have just shown, like this web process for el profesión?
Yes.
Is it possible to use your models to like fit my data and like gain more insight into that kind of stuff?
So, is it possible to use the model to fit? So, currently our SDK actually you're able to use the model. So for each of our data set, you can use the model so that you can integrate in your own system. The code that is the fitting code isn't part of this release. So our modeling team is still currently working on how to refining that fitting code and also making that available to people. So the short answer is no at this point in time, but I think the intention was that one day they will release the fitting code as well.
Yeah, how about like getting the models? Like what type of format? Do you have any like what type of format are the models available?
So there is, I think there's an entire section of the SDK just devoted to models, and this is what drove us to. We know that we had to do an SDK because of this. So models.
Do you have questions whether you abstract somehow or like different languages simulators? Sorry, no, I guess my question is like, well, okay, are you abstracting somehow over different like languages and simulators that can be used?
Neuron, the big physics neuron or neuron.
Sorry, what?
Neuron. Yeah.
But, I mean,
Get down
and
to
that's
neuron.
the only language you
All Python code for the glib.
yeah.
So I think currently they, it's interesting. So for the neuron, there is as part of this code is that they try to write a Python wrapper on top of the neuron. So basically, we have what you're downloading is the parameter file, and that parameter there's Python code to expand that parameter file into something neuron understands more natively. So with our data, we often welcome people who mash up our data with their own data set. So the biophysical model is also in ModelDB, in which case they said, well, all their stuff is all in native neurons. So they took our Python code, they make that available, but they also expanded it to more common neuron format as well. So there is, in our modeling team, there's every attempt to try to have that layer of abstraction from the underlying model, but that's just it's work in progress because of the different levels of modeling that we're trying to use. Have anybody actually used either the connectivity database or the cell types database? All right.
Actually, sorry. When we're talking about cell morphology for specific genotype types, are those representations fairly consistent across cells? I'm just like, it probably relates to what I asked before, but so if you're looking at a given cell genotype, let's say like PV expressing cell, are those morphologically similar to each other?
So currently, so I think with the current transgenic technology we have today, I think you should view that as its ability for us to sample a laminar population. Within that Cree line, we also know it's a mixed population. So sometimes you would actually see different cell types within one Cree line as well. So the hope is here is that we collect all this data using our first set of Cree lines, and then we actually take all the data and then do a classification on that. And this is why it was important to also connect our data to transcriptomic. So right now we take the data, we do electrophysiology that gets filled with biocytins so that we can do the morphology. But what we want to do is also add another part of the pipeline in which enough material is then sucked out of the cell so that they can do transcriptomic as well. So if you have this framework and then you find that even with this Cree line, you're seeing different population because the morphology looks different, you can then see what gene marker is would then well, the set of genes that actually would then was able to then divide the Cree line up into further subtypes. And then the hope is then we'll make a very specific Cree line, and then if we use that into the pipeline, then everything should be very stereotyped. So currently it's not, and we knew it wasn't, but part of this project was the ability to get to that point.
But presently the data that are published already on the website with morphology as it relates to, like, say, input resistant, the type of model that the cell applies to, those are just some examples of the cells. Those are not like...
Oh, okay. So, so the intention was to reconstruct as many cells as we can. The reconstruction process is actually only semi-automated. So, as each data set, each cell will receive, there's an assessment of how good the biocidin fill was, because sometimes that the fill doesn't work out well. If the fill works out well, then the data would then get imaged for that high resolution stack, and then they will be traced for morphology. So because of that, not every data set has morphology. So if we go back to here, let's clear, reset. Let's reset everything. So in the entire data set, we have actually 318 cells. And then about a quarter of them have morphology. So it's not example as much as we can if the data allows for the reconstruction. All right, I think I'm going to hit one slide here. All right. So then looking forward in the next couple of years, as I said, that we want to integrate PatchSeq into the data framework as well, so that we can then have to create a taxonomy that includes electrophysiomorphology and transcriptomics. And as Christoph mentioned, that we're also starting to integrate human data into the pipeline as well. So in next year, as well as seeing mouse cells, you start to see human cells as well, and we will run it through the same pipeline to get that same information, and then they'll both go in the database. And again, you would want to do a taxonomy, using the mouse data on its own and using the human data on its own. And then the interesting part is then to see whether to contrast and compare between the data and its organization. We're also working at doing multi-patch data. So currently we just patch one cell, we do a recording in one cell. When we do this multi-patch, when we have a multi-patch working, the idea is to then patch more 4 to 8 cells, in which we will then inject current in one cell to see the response in the other cells. So as well as getting a characterization of its intrinsic property, you can also then know which cell it's connected to. So with that data, we're going to slowly build up a sort of a functional connectome of that data set as well. And in our next release, we're going to have more single patch data. We're going to expand from the ALM into the LGN as well for the single patch data. On the website, there's also currently there is a 1,700 transcriptomic data for the LGN. In this release, we're going to have additional cells from AOM, which is the motor area, and also from V1 data as well. And then I've also talked about the human tissue as well. It'll be interesting how we deal with human tissue, is getting things oriented and then relocated back in a common framework is a challenge that we need to we need to put together is because not only do you want to know where the cell comes from, because it's surgical tissue, it can come from different places, and I think that's an important metadata for those cells. Okay, I think this was okay. I'm going to switch over to the cell types database. All right, so then in June this year, we're switching over this to the Brain Observatory. In June this year was our launch of our Brain Observatory data. So the idea here was that what we wanted to do is to fill out this data cube in which we go and survey the different cortical, the different visual areas at different depths using Cree lines using Cree lines for the sampling, and then show the mouse various different stimulus, and then using that data to then characterize each cell, and so to give it a functional characterization. So this is probably the most complicated production pipeline we have to date. Before that, we had a single brain. It gets cut up, it gets imaged. This is the first time we're dealing with a live mice in which multiple things happen to it, happens to it repeatedly. You'll keep track of that mouse and then keep track of all the cells as well. So it starts off with a cranial window being put on the mouse. We then keep track of the photodocs of the vasculature, because you want to keep track of the health of that cranial window, and that's preparation. And the first thing that happens is intrinsic imaging, in which we're going to map the multiple visual areas. Here we have to keep track of where everything is with respect to the vasculature. So every time we say we compute a bit of information, we make sure that we can always overlay that to the vasculature. And then our engineering team has actually put in a cranial window coordinate system in which every one of our different system is then calibrated. So then in our intrinsic imaging system, if you're interested in making an injection here, then our system is then calibrated. So then when you go to our calcium imaging rig, you can type in the coordinate system so that you, as a starter place to where you need to look at, because they're at different scale. Because we're doing live mice, the mouse needs to be habituated so that it's then relaxed and able to complete its task. We do the calcium imaging as the mouse runs on the wheel, while the big monitor shows the different visual stimulus. When we collect the data for every session, we keep track of the calcium imaging data. We keep track of the eye position. We're still working on how to in an automated fashion, find the pupil so that we can then, for every time point, also give you a coordinate of the screen where the mouse is looking at. And then we also keep track of we also have a video of what the mouse is doing, so that will actually give you a behavior annotation. Is that a question?
How stable is the vasculature over time? Is it nanometers, micrometers, centimeters, with respect to the rest of the cortex? So if you look at one vessel in my brain today and tomorrow, is it moved by?
It does move. We can't map it precisely. I think is orders of micrometers. You have your big vasculature that doesn't move, but the little ones, you may not find them again. Yeah. And I think that's also part of our the animal gets a photo doc regularly. That's part of our QC process as well.
Isn't the data from John Reynolds' lab and others doing optical imaging, where they look at the window and the vessels have been moved and shifted?
Well, that's the question, how much and
They can
what types
shift.
get, so the key segments might be.
Yeah, I think they claim larger.
Yeah.
But of course, you don't have Dura there.
Is it like ISOs?
No, that's not the stack.
And
We're talking about weeks, you know, so
Yeah.
the imaging takes place for 2 to 3 weeks in a mouse. And so you can use them as landmarks to come back to this thing cell. Who knows what happens over a year, let alone in 1 week.
so yeah, because we're over weeks, we're hoping that the change isn't that much, so that we can still keep track of everything.
Your habituation is 7 days, right? Or about 7 days, as I remember, and then you report in graduates. So it's like, it's a very short period of time.
Yeah. Because they're not, yeah, because it's passive viewing, they just need to be able to be okay to be on the treadmill. In this first data set, we also have done a second intrinsic imaging data, just as in our first data set, just so that we are able to compare back to the our first data set, just to get some sense of how much that changed over time as well.
For the task, do the animals have to run in order to get a reward or whatever? I couldn't quite.
I believe the habituation is so that they would run and be comfortable with this task. For the passive viewing, they don't have to run. In fact, I think during the imaging session, they don't lick. They don't even lick. During the session, they can choose to run or they can choose to stay still. So there isn't a task component to this part of the project. So as the next part of the project, when we're wanting to use this data to start to understand behavior, then there will be a task in which they need to do something or stop or lick or press a pedal or something like that. So that's to be determined what that paradigm is.
So right now that happens in research science, so we do training and rewarded, all of that, but not yet in the pipeline. I think that's the goal for 2017. That we have vigilance, so we know the animals is vigilant in behavior, and then in particular, we are focusing on object recognition.
So then the last step for the mouse in the pipeline is to actually go through zero true photon imaging. It's the same system that we use for the connectivity data, so that we have this nice 3D model of the brain. That's important because with this big data set, what we're looking for is in our volumetric data set is surface vasculature. So we have to have good imaging and also not a whole lot of deformation, because then we're going to take the data set and just look at the surface, just look at the main vasculature, and that's how we align the intrinsic imaging data to our CCF through vasculature, and it's also through vasculature that each of the individual calcium imaging data is then aligned to the ISI data. So we're all about the mouse vasculature in order to keep everything coherent in this data set. We're still working on this part of the pipeline, because you can get information about cells here, but then the next step is to then give it assign each cell to the framework as well. All right, Christoph already showed this. We've done this already. Again, this was extremely important in order to make the experiment work, because not only were we going, not only did we want to return to the same place, it was important to us to target particular retinotopic locations within all the different areas. So we had to look at the sign maps, pick the points that we want to go to, and then have to rely on the coordinate system to help drive us to those points. All right, so that was all the complication just in data production. Then there is the data processing. So for the intrinsic imaging data, there's processing to generate the sign maps, then to do a segmentation of the sign maps, And also an important part was we wanted to make this automated as possible. So once it's got the segmentation, there is a part of the pipeline in which it would try to assign it to one of the visual areas, given that it's been trained to understand the properties of a visual area, which sign it is, who it can be next to. Even with that, we had to make a UI so that an operator can always override our annotation, and also override when the segmentation, when the boundaries are sort of a little fuzzy and we didn't get the segmentation correct. So there's that process in which we have a segmentation annotation QC. Then once that is done, then we make targeting maps, because we want to target. So we want to target the center of V1. And then we have to find the corresponding retinotopic point of that center in all the other different visual areas. And then we create a map so that the operators can then drive to those locations. And then for the calcium imaging data, once we receive the movie, lots of different processing has to happen. Firstly, we need to do motion correction for that data set. A lot of engineering work was done to try to get rid of the Z motion. Multiple design of that head frame was done to reduce any brain motion that happens as well, so that we can just try to correct for XY motion. And then once we've done that, we do segmentation to find cells in the images. Segmentation, we went old school. It's old school segmentation in which we take the entire the movie and then broke it out into chunks, into small chunks. And each chunk, basically, you're making a max projection image, and with that, you use basically old school adaptive thresholding and sort of morphological sort of filtering to find round things in each of these images. Then you need to stack everything back up together to say, because we've divided this up, we actually find two objects, but they're actually the same cell. And then so you can stitch all of these things back together again. But now, because the data had to go through multiple sessions, you have a segmentation mask for 2 different sessions. And then again, we have to do registration of the 2 sessions, because they're always slightly out, and then try to match those cells again, so that at the end of the day, we have this concept of this one cell and which pixel, set of pixel it belongs to in different calcium movies. All right, so this registration cross signal sessions, then we do extraction of fluorescent. We have to do a neuropil subtraction, because if you just do a straight time series from your ROI, some of that part of that signal is actually from the neuropil and surrounding areas. So then now you've got to make a determination what pixel around me I can use as a model for the neuropil signature, and then that is then divided back out from your ROI. And then right now we're also trying to work out demixing, because when we do segmentation, because we divide it into chunks, as long as 2 cells are overlapping, as long as they fire at different time, we actually can capture them as 2 different objects. But then we now need to demix the signal as well. So currently, when the cells overlap, the signal is mixed, so we're trying to unmix that as well. And then this is now, we have, to drive this experiment, we have multiple different system running on its own little clock. So we have the calcium imaging, that rig, it has its time clock. We have the stimulus workstation that has its clock. And then we also have each of the cameras that take pictures of the eye and of the body image, that they have their clocks. And then when we do task and reward, then we have the licks, which then needs to be logged as when the licking has happened. And there is the encoder that keeps track of how much the mouse has run, because that's also a behavior. It affects it affects the signal that you see. So all of these clock goes into a one machine that logs everything, and then part of the other process, part of alignment that we have to do is to synchronize everything into synchronize everything. So when we package these things up for you in an NWB file, everything's already synchronized, and then you can just use it as time series without having to figure out, you know, the different rates and the different timings of the data sets. And then, as I already said, and then the next step is to try to synchronize everything spatially.
So one quick technical question about the segmentation business. Compared to, say, a human observer tracking frame by frame, as boring as it sounds, one cell, how accurate is this? I mean, when you say it's, you make one mistake every frame, every hour, every day, every year.
So we qualitatively have done that. We want to do that more quantitatively, but then it's actually hard for a human to trace every cell, because often they will do it in the max projection image, but that sometimes you have a cell that fires lightly and then it's hard to draw that, or when you have cells that are overlapping, it's hard to draw that. So we're still having issues. How do we even have that ground truthing in order to do?
So what is ground truth? If it's not a human observer, what's ground? Is there another way to define ground truth?
So I think the ground truth is to have another data set in which now you do a triple transgenic, so that every cell has some sort of red channel marker. I think that would be the final truth of, did you find the X number of cells? So we don't do that here because doing that 2-color imaging is very expensive. Even with just a green channel, each data set's about 70 gigs on its own. But I think what we need to do is that for a certain validation set, that I think that would be the final ground truth, that you have to match it for each red soma that you see. Did you detect a signal? Because even if the cell didn't fire, then that's important information as well, to know that there was a cell there that didn't respond to a stimulus at all. Right now, we can only segment cell that had a response to the stimulus. So I think we also need to know.
The entire session?
Yes.
That are cells that never fired an entire session?
Yeah. There could be, right? We don't know, because we can only find things that are actually activated. All right, and then we also, another thing we need to do more deeply is that these Cree lines, while it's essentially meant to be the same Cree line as what we're using in our connectivity data set and the cell types, to put the, again, I'm the software engineer, to enable the GCAM. These are actually triple transgenic, and it gets crossed with also CAM kinase as well. So I think we're still trying to do a very good characterization of which cell population does actually have the GCAM signal in it as well. So I think it's a mixture of what our original Cree line is, but then that CAM kinase sort of comes into the mix as well. So then I think we want to do our characterization study and also publish that as part of the product as well.
Also related to tracing individual cells, how stable is expression across time? So if you were to trace a single animal for like several weeks, would it be the case that sometimes like GCAM expression would appear in a given cell just based on like developmental Cree expression and then like go away, and this would mess up your ability to detect individual cells?
I can't answer that with full knowledge, but I don't believe that's the case.
I know that's an issue with rodents, with rats, where you express GCAMs virally. I'm not sure if that's an issue with breeding transgenic expression.
Okay, so that's a question I can't answer. I can find out, but I can't answer directly. Well, Christoph, he might be able to answer that, but he stepped out. All right. Okay, so then with all of this work, then we'll end for our release in June. We ended up with 75 experiments. And an experiment is when we have successfully, in one location, successfully the 3 sessions, and everything has passed QC. So it only goes out to the web if we have done all the sessions, because we needed all the stimulus to actually so that we can integrate and do our computation. And then in our database today, we roughly have 18,000 cells in which we have done some quantification to look at some its characteristics. Okay, I am going to go here. Let me. So I.
You can control click it and
I think if I did that and went like that, no. Aha. Okay. All right. So then, this is basically for every cell gets this summary page, and so the visualization is something that you guys probably haven't seen because we sort of invented it for this product. So for this diagram represents basically response to every trial of the natural scenes. So we had 120, I believe, 128 natural scenes, 118 natural scenes that were repeated multiple times. And then we want a way to be able to show all the variability of every trial in a single diagram. And we knew we couldn't do that with a time series because there was just too many time series to show. So what we did was that for each trial, we basically compute df over f over that trial, and then we arranged the images along a circle. So starting here is 0 index image. As we move along, there were different images that's presented to the mouse. And then along the ray, we wanted to then, as a heat map style, to put a dot, and the color of the dot represents the strength of the response. So when it's very dark, it had a very strong response. When it's very light, it has a low response. And then we then ordered it such that the highest responding trial is towards the center of the circle, and then it fades out. So in a view like this, you can at a glance see that this cell is highly responsive to the image of the butterfly and also of the bird. But it also shows that it doesn't always fire. Just because the butterfly appears on the screen, there are times that it doesn't fire as well. So this was the ability for us to actually give a good summary of what's happening to the response of every trial in our natural scene presentation. The length represents, okay, it's the same length.
It's just they get
I think otherwise means that there are many cases where it doesn't factor.
Yeah.
Where there's no activity, that's the number of trials. So they all have the same length.
They all have the same length.
We just don't see it because a lot of cells don't care about how much it's not light. That's
And then we had lots of conversation of why did I have to put in a circle, why couldn't I put in a line? But actually the circle is nice because when you, for example, if there was an image here and if another cell likes the image here, when you put in a circle, there's enough of an angle that you can tell the difference. But when you line it up, you then have to look very carefully if it really is the same image. So then we did the same thing with the drifting gratings. We actually have 8 different orientations, and 1, 2, 3, 4, 5 different spatial frequency. I really like this viewer because at least you can see each of the stimulus as you hover across, and then at a glance you can see that this cell is direction selective, and then it's versus being orientation selective.
This is the same cell.
This is the same cell.
Oh, so if we go back to the image, the monarch butterfly and the whatever it is, the masked booby bird, it's going to be like there are vertical lines.
And yeah, so I think one of the things is that the way the monitor is set up, they're looking at something over here. So the receptor field, I think the receptor field isn't very as strong for this one, but typically receptor fields are here. So during the summertime, NeuroFutures, we held it at the Allen Institute. So if you go to our YouTube channel, Saskia de Reeves, she started to show some of the analysis that she found for this data set. So some of the natural images, I don't know if I can find it quickly, of a set of pencils that were facing up and another set of pencil that was horizontal. And then so because we have so many cells, she can start to see relationships between gradient information in the image and also the drifting gratings as well.
So these are baby mice. They're awake.
They're awake.
Yeah, and so you tracking eyesight movements as well?
We do. We've got the movie. We're just still trying to work out how to do that robustly.
Okay, so how much do they actually move their, I mean, you're actually receptive fields.
So
You must have enough data to track their eyes, that they're,
yes.
they're not trained to fixate.
No. So that's a very good question, and that's also speak to why we're having such a difficult time with tracking, because well, firstly, they can do whatever they like. Sometimes they sit there and they whisk, and then all of a sudden, our movies suddenly all change shapes. They do, the eye moves quickly as well. So I think that's could be vital information to explain why sometimes it fires and sometimes it doesn't for the different. We do have running speed.
Some people have claimed that depending on the running speed, there is more or less activity.
So with the help of Saskia, we made a graph for the running as well to see how tuned a cell is to the running.
Also explain some of this variability potentially. Is they may be running faster than
Yeah.
others, they may be running
Yes. So then it will be interesting. Well, it'll be interesting when we have that information, and if running in eye position a key signature, how we integrate that back in to here would be an interesting visualization challenge. So here is to respond the same cell to static grading. Here the stimulus just flash. So because it likes this direction, therefore it also likes this orientation. It also is more responsive to a lower spatial frequency than a higher spatial frequency. Why are there 4 little patches here? Because it's just flashed, and because they're worried about the receptor field, and because some of our grading is kind of fat, they've actually basically moved each of the grading in 4 as well, to make sure that it was just because you hit the white spot of that grading, it wasn't driving what you the signature, and that's why every one of these have 4 little spots, and some phases are actually more responsive than other phases.
Are these flashed so fast so that the mouse can move its eyes while the image
This is where I need to then.
I think that's slow.
Wait a second. Okay, here we go. It's presented for 250 milliseconds.
So, okay. From what we learned earlier today, that's long enough for it to staccato a couple of times. That's true.
All right. Okay. I like to show it here because then I can move things around. Okay, these are the movies. So the Touch of Evil. We showed the same 30-second clips. We repeated in every session so that we can then. It's the only thing that we can compare two sessions together. So then this is, and then what we've done here was then we divide the time up and then we computed a DF just like we did for drifting gratings and static grading. And so each of these rings represent one trial. So for each clip is played 10 times to the mouse, and the blue ring at the end is the average over the 10 trials. We weren't fair use meant that we couldn't play the movie here. So what we did was then we chunked it into certain chunks. So then as you move around here, you are able to see at what scene that the mouse is or the has a strong response for. So again, it's in a circle so that it's you can easily compare the cells to the repeats to each other and also against other mice as well. So this one has a nice repeat in 1A, B, and C. Movie 2 is not structured because I think all the motion is actually very different to the first clip. And then we also showed a 2-minute clip. This is why things, and when you see response is really tight because it's a longer clip, and then we've quantized that. All right. Okay, I have to go back to here now. All right, our locally sparse noise. So then we have this locally sparse noise stimulus in which approximately each frame there's 10 black or white spots per frame with a 20 degrees exclusion zone. So if we, the premise of this is then every time, for example, for here, every time there's a white square at that location, we can just treat it as a trial, regardless of what's happening all around it. Because on average, if you're only in the context of this square, every other position on average will just all gray out. So basically then we're treating each of it as independent trial, and just like the other plots, for each of the 16 by 28, it's a little sort of a little circle in the middle in which again we sorted out each of the 115 trials so that the highest response is in the middle, and we do a circling of that out. If we made this, if it's actually smaller. So we need to tweak this a little bit. In some of the cases, you can actually see the receptor field because it was more consistent, that you can see a stronger representation here. The idea of that is also we do it for the off trials as well, so that you can do a comparison of on and off subunits. All right.
So it's not distinguishing between black and white in that case?
Sorry?
When the spots are presented in black or white, it's not distinguishing.
It's not. Yes. So this cell really, it was just responding. Yeah. Yeah. All right. So, and then these plots are meant to attempt to show tuning to running. So the blue is, and we also divided the whole data set into into sections in which there was no stimulus. So for each of our sessions, there are periods in which just a gray screen is shown. And so this is then for between the periods of time when it's just a gray screen versus a period of time where there's a visual stimulus, and the axis here is how fast the mouse is running. So then when there's visual stimulus, this cell has high response when it's running. Interpretation of this graph so that we can have some sense of how tuned it is. So there are graphs that goes up and there are graphs that goes down. And then we have 3 of them because for each sessions they are independently quantified.
So these are, this is a lot of data from one cell from one mouse, right?
Yes.
So I wonder, is there some systematicity? So where are these being sampled from and how densely?
Okay. Let's go back to the main page. I'm just going to get us there. All right. So each field of view is it's 400 microns. So then typically, for we're getting 100, 200 cells per field of view.
Is there some systematicity on the neurons that you're sampling from? So do these properties vary along different units that you're reporting from a single neighborhood?
So we're collecting the data. Analysis is coming. So here is our first attempt to basically try to tell that bigger picture. Currently, when you click on this, you get all the data. We're working out what's the best way of filtering. So then each row represents basically one of our experiments. So we have, we know which area this is, we know what query line it comes from, what imaging depth, and then here are basically a summary of what that population of cell did. So here is basically a cumulative histogram of the orientation selectivity, of its preferred spatial frequency, for things that, for the static grading, we also computed this time to peak in which the stimulus is shown, and then how long did it take from the stimulus to actually reach its maximum response. Then for the drifting grading, again, a histogram for direction and orientation selectivity. So then right now you're scrolling through this. So what we want to do is then when people download the data and when we do a bigger analysis, then we can say how stable are these populations. So when you are looking at one experiment, you can click on this thing called View Cells. And basically what we're doing is we're trying to put all the thumbnails in a single place. I'm just going to make this a little smaller. Okay, you see when we couldn't reliably find the same cell over the 3 sessions. Is because sometimes if that cell in that session has moved too far away so that we can't reliably say it's the same cell, or it wasn't responding in that session and we had to make up that there was a cell there. And so you can then, for example, so for this experiment, let's see, I didn't pick, or I didn't stage this, so let's see what we get. Show filters. Okay, let's start with the static grading, and let's go and find cells with, well, it's easier in drifting grading, so we go. Drifting grading. Drifting grading, let's find cells with particular preferred orientation. So here we go. So here I'm setting it with a preferred orientation of 0. So then you start to see that some of the cells have, it's pointing this way. You can then go, let's find some cells that are at 45 degrees. Now only filters cells that have a certain orientation, direction index, so that you can further find cells that meets your. And then for, well, let's clear all the filters. Clear filters. And then for the natural scene, you can go in and say, whoa, well, no. For the natural scene, you can go in and basically start to walk each of the different images to find images that cells that prefer that particular image. Right now, we haven't got an index to say it likes that image more than every other image. It's just whatever is preferred at this point. So here you can see there's a spike here, and there's a corresponding spike here to the image 3. And once you find a cell you like, you just click on any of the thumbnails, and it will get you to the big details page so that you can then look at the thumbnails in greater details. Has anybody actually looked at this or played around the data yet? So we will be at SFN. We have a booth there. So when you guys play around there and when you got questions, I think these questions are going to be complicated questions. We'll be there, the scientific team, the technology team, so that we can help answer some of the questions there as well. So I think we don't have a systematic way of doing that, but I think the best thing to do is that. So the best thing to do is there's a great big button called "Send us a message," and on the other side of that is our application scientist. And if you say, you're interested in this, what she would do would basically then basically sort of hook you up with Michael and Saskia, who are doing some of the main analysis for this data set. So for each of our big data resource, we also have this concept of the platform paper, in which because it's still at the beginning, is basically a bunch of vignettes describing how we did this, but is also little scientific vignettes are required also for that paper as well. So it would be something that we would do there, but we're still trying to figure out how this integration works as well. But I think talking to Michael and Saskia is a great first step as to how to do that. All right.
Any other questions for Lydia? All right, let's thank the speaker.
Thank you.